Okay, good day, class. Now we are on the final part of your innate immunity 2 or innate immunity series lecture 1 and 2. So we started with your 1, okay, discussing the humoral part. We discussed the inflammation. We discussed the activation of your APRs by your cytokines, interleukin 1, interleukin 6, and your TNF to your liver, then APRs 10, tadan, inflammation. And then we also, part of your inflammation is the migration or calling of your neutrophils to the site of infection. And then we move to your immune cells, which is the first part of your innate immunity part 2. So as I mentioned in the previous lecture class, that the main function of your immune cells, but not all, is for your phagocytosis. So for our topic for this video lecture, we will now try to cover your phagocytosis. Okay? So this video lecture is kind of simple, so I hope that you understand the concept because the for the preparation of your quiz exam and also for your board exam. Okay, again, I am your professor, Alfredo Jr. You can call me Sir Red, and if you have some questions with this video lecture, you can contact your professors so that we can clear up the gray areas of your immunology and serology. Again, you can follow my video lecture with the reference book. You have your Stevens and you have your Turgeon. Okay, so let's start with the discussion of your phagocytosis. So, as I mentioned earlier, that your phagocytosis is still part of your innate, okay, immune response. Okay, the reason why cells of the innate immune system are considered to provide a non-specific immunity is due to the nature of the receptors. Okay, so we have different receptors. If you can observe on the on the slides, you have your mannose receptors, you have your TLRs, you have your dectins, and you have your lectins. For your dectins class, okay, your dectin, although we will not thoroughly explain this one in your basic immunology and serology, but when you, for example, take your masters, we'll discuss that your dectin is associated with your fungi. For example, your alpha mannans and your beta mannans. And then your dectin 1 will identify your alpha mannans, your B mannans will be identified with your dectin 2. However, in this video lecture class, we'll discuss more on the bacterial part and then also viral part with the use of your TLRs or toll-like receptors. Okay, cells of the innate immune system are able to recognize a distinctive molecular structure. We call that PAM class or pathogen associated molecular pattern or PAM. These are distinctive structures that are synthesized by foreign pathogens, and we'll try to discuss that one on the next slide. Okay, so I just wanted to like show to the slide. Okay, allow me to have my sorry class. Allow me to have my pointer. I just wanted to show here, class, that this is your cell, and each cell has different number of your TLRs, dectins, lectins, and mannose receptors. These are responsible for identifying, okay, sila yung responsible, class, how they recognize that this organism or this foreign, that this pathogen is a foreign, not a self-antigen. Okay, so this is due to your TLRs, mannose receptors, dectins, and you have your lectins. For your toll-like receptor, we'll focus this one. Okay, your toll is a protein originally discovered in the fruit fly, okay, Drosophila, okay, where it plays an important role in antifungal immunity in the adult fly. However, they also found that this toll protein is also found in humans. Kaya, they term it toll-like, okay, receptors. Okay, very similar molecules are found in humans, okay, leukocytes and some non-leukocyte cell types. Then, therefore, they are called a toll-like. Okay, kasi originally they are from the fruit fly, toll protein, and then they found in humans similar structure, so they called it toll-like receptors or TLRs. So what's the function of the TLRs? Okay, so your TLRs or toll-like receptors are found on your immune cells, or any other part of your cells also have this one. But the highest concentration of these receptors occurs on your monocytes, macrophages, and neutrophils. So what are the basic common of these three immune cells class? Correct. They are phagocytic cells. Okay, phagocytic cells usually have their toll-like receptors. There are 11 slightly different TLRs and humans. However, in your book, if you can observe here, this is from Stevens. You have your TLR1, TLR2, and TLR4. Okay, for example, your TLR1 is specific for identifying mycobacteria. Your TLR2 is specific for identifying your gram-positive bacteria. Your TLR4 is for cell membrane. Your TLR4, sorry, class is for your gram-negative bacteria. Okay, but I wanted to also explain, class, that this is not only limited to the three TLRs presented in your book. We have different TLRs, class. Like for example, your TLR1 and your TLR2 are specific to your diacetyl lipopeptides. Your diacetyl lipopeptides, class, is your LTA. It means, class, that if the bacteria or the pathogen contains an LTA, LTA stand for your lipoticoic acid. And we know that lipoticoic acid, class, is part of what organism? Correct. Gram-positive cocci. So therefore, those organisms under gram-positive cocci, they will be detected using your TLR1 and you have your TLR2. Then we also have your Tri-Acetyl Lipopeptides. Your Tri-Acetyl Lipopeptides, okay, those are a component of your bacteria. Example, sir, kasi mahirap naman intindihin ng Tri-Acetyl Lipopeptides. Ano yung mga example niyan? Okay, one best example of your Tri-Acetyl Lipopeptide class is your, correct, you have your peptidoglycans. Okay, peptidoglycans are present with your gram-positive organism, also with your gram-negative class, but very small. Okay, compared with your gram-positive organism. Okay, so sino yung ma-activate and or magka-catch with your peptidoglycans class? You have your TLR2 and you have your TLR6. So sir, how about gram-negative bacteria? So the gram-negative bacteria contains LPS, lipopolysaccharide, or lipid A. Okay, we learned that one already in your bacteriology. So, ano yung class? Ano yung receptor that can identify your gram-negative bacteria? So, you have your TLR4. And also, those organisms that contains flagella, okay, take note, class, that the basic unit of your flagella is your flagellin. For example, your pili, the basic unit of your pili is your pilin. Okay, so for your flagellin, okay, for flagella or flagellated organism, then you have your TLR5. So example, you have your E. coli. E. coli is a peritrichous organism. Ibig sabihin, class, that, for example, this is your E. coli, this is your flagella, this is peritrichous. Okay, so ibig sabihin ng peritrichous, class, the flagella is located around or covered around with the organism. So this is your E. coli. We also have atrichous. Ibig sabihin, class, walang flagella. That's the reason why meron siyang affix na ibig sabihin absent. Okay? So, example, you have your Shigella. Okay, Shigella is non-hemolytic. They are non-motile, okay, organism. So, therefore, hindi ma-activate si flagellin, hindi ma-activate si TLR5 because your Shigella does not contain your flagella or flagellin. We also have your propylene for TLR11. You have your double-stranded RNA for viruses. Okay, you have your TLR3. We also have your TLR7 for your single-stranded RNA. Those are viruses. Then you have your single-stranded RNA also. You have your TLR8. And then you have your CpG DNA. You have your TLR9. You're familiar with CpG already, correct? Because I believe that you took your, I don't know if you took your molecular biology yet or not, but majority of the bacteria, okay, class, their DNA are CpG coated. Okay, so it means that your TLR9 usually will be activated once the organism will be engulfed. So once a receptor binds to its a particular substance or ligands, okay, phagocytosis may be stimulated. Okay, thus the play an important role of enhancing natural immunity. Okay, maybe before in your human anatomy class, you discussed phagocytosis already. And then you learn that the phagocytosis, siguro simple lang. Yung pagka-discuss, ano lang, initiation, degranulation, and then egress or release. Yun lang, diba? Pagdating sa immunology, let's try to kind of elevate what we learned during your human anatomy or during your high school years. So your phagocytosis is actually divided into two. Okay, so they are not divided, they are classified into two. Either the phagocytosis is direct or indirect. So when we say direct phagocytosis, it means that they don't need a mediator, okay, or a third party. Just like, for example, in this picture, you have your antibodies and you have your complement fragments. So for direct naman class, no need for opsonins okay, via primitive pattern or PRR and then to your toll-like receptor, then engulfment. With the indirect naman class, it requires opsonization. So sir, anong ibig sabihin ng opsonization? Kay opsonization, class with the indirect and direct phagocytosis, opsonins is to prepare for eating. Tagging class. Okay, the other term of your opsonin is tagging. So it means that when you tag someone, so that means opsonin. Okay, one best example of an opsonin is your antibody. Kasi your antibody, di ba, as I mentioned before, that your antibody is not capable of destroying the bacteria, viruses, or etc. They don't have the capacity class. Okay. However, what they usually do is to bind and then expose their butt, which is the FC portion class. And then the FC portion will bind to the phagocytic cell. And we call that one indirect phagocytosis. Indirect because it requires a opsonin for them to detect the organism for phagocytosis. Compare with the then phagocytic cells have receptors for antibodies and for complement components which aid in contact okay, and initiating ingestion. So, ayan, nabanggit ko naman kanina class that the antibody will serve as a tag together with their complement components. Compare with your direct naman class, okay, with your direct phagocytosis does not require the opsonin. Then therefore, the bacteria together with the TLRs, or we call that one PPRP, primitive pattern recognition receptor, or we also call that one PAM, or pathogen associated molecular pattern. Okay, recognize carbohydrate and lipid sequences in your microorganisms. Like for example, you have your LPS, di ba? Ito. Allow me to go back with a previous slide, class. Here, this one, LPS, flagellin, you have your Tri-Acetyl Lipopeptides, peptidoglycan, LTA, lipoticoic acids. Those are your pumps. Okay, pathogen-associated molecular patterns. Okay? So, yung partner ng pathogen-associated molecular patterns mo, class, is your TLRs or toll-like receptors. So, mag-partner yan sila. Okay? Take note with that one. Kaya pagdating sa quiz exam and board exam, what you need to prepare is that with this type of EPAM, pathogen-associated molecule, which of the TLR will be activated? So for example, I have a peptidoglycan. Which TLR will be activated? If I have a LTA, lipoticoic acid, which TLR is activated? So you need to memorize that one. Okay? So in the process of your phagocytosis, it consists of 4 main steps. Of course, class, having problem with the... Okay, so here, it always start with the physical contact, okay, between the white blood cells and the foreign particles. Okay, with the physical contact, it either be direct or indirect. So it depends if it requires the opsonin antibody or your complement fragments. So I think in the case of this one, it does not require opsonin, so direct phagocytosis. Then after physical contact, then now formation of your phagosome. Okay, your phagosome here will try to cover or engulf inside your bacteria. Okay, because your phagosome have a mechanism in killing and then it will release your lysozymes. Okay, so the fusion of your cytoplasmic granules to form a phagolysosome. Okay, so after the formation of your phagolysosome and then your phagolysosome, okay, so that's your phagosome plus your lysosome, yung blue one, this one, the small blue one class, if you can observe, these are your lysosomes. Then you have your phagosome. So if the lysosome will fuse to your phagosome here, then they are called a phagolysosome. And the function of your lysosome, as I mentioned that one during your phagocytic cell granules function, is to degrade your organism. And then after degradation, of course, it will be a digestion and release. So this is a basic steps of your phagocytosis. So let's go back with your inflammation. So as I mentioned earlier, not earlier class, as I mentioned with the previous video lecture, that your activated macrophage will release your cytokines, and your cytokines will tell your endothelial cells to release your selectins. And your selectins will bind to your selectin ligands from your neutrophil having rolling, and it will have an expression of your integrin. Integrin is very important because it will bind to your ICAM1. And then your ICAM1 will try to pull it out to the lumen of the venule to the connective tissue. Okay, so after this one, we call this one a process diapedesis. Ibig sabihin ng diapedesis class is the movement or physical movement of your neutrophil from rolling to demigration. So we call that one diapedesis. Okay, this process is aided by your chemotaxis. Example, you have your cytokines, interleukin-6, and you have your TNF-alpha. Okay, so please take note, class, that chemokines or chemotaxis are the communication molecules of your cells. So without the chemotaxis class, the movement is random. So example of your chemotaxis class, we have positive chemotaxis and you have your negative chemotaxis. So ang ibig sabihin ng positive chemotaxis class is, so for example, this one is your cells, then this one is the other cells. So if this cell will try to release chemotaxis with this one, and tapos positive siya class, what will happen? The other cells will try to go here. However, pag negative chemotaxis siya class, and then this is the cell, the cell will release a negative chemotaxin. Tapos kung magre-release siya ng negative chemotaxin class, the other cell will now detect na, okay, hindi ako kailangan dito, so I need to go out. Okay, so yun yung negative chemotaxin class. Okay, so take note with the difference of the two. So again, so example of your chemotaxin are antibodies, complement fragments, you have your CPRs, you have your bacterial factors, chemotactic cytokines. And as I mentioned, that your chemotaxins with your chemotaxis will serve as the communication molecules of your cell to cell talks. Okay, so paano sila nag-uusap? It's because of, it is due to your chemotaxins with your chemotaxis process. So therefore, class, pag tinanggal natin yung chemotaxis class, the cell will become random. Di nila alam kung saan sila papunta. Okay, so therefore, your chemotaxin with your chemotaxis is very crucial with your cell-to-cell interaction. So, with the process of your phagocytosis class, we identify already that the initial phase is with the recognition. After recognition, then the second phase is with the fusion of your phagosome to your lysosome. Then we'll have your digestion. With the digestion class, we call that one cytopepsis. Okay, we have two mechanisms of your digestion. First is due to the release of your granules. Okay, your granules in the phagocytic cytosol migrate, fuse with the phagosome to form a phagolysosome. With the phagolysosome class, with the degranulation of your neutrophil, it will lead to the release of your lactoferrin. Okay, your lactoferrin will bind to your iron. We discussed that one in the previous video lecture. You have your lysozymes that will target your bacterial cell. And then you have your defensins, can increase the membrane permeability. And then you have your elastase, an enzyme that will damage your host tissues. So again, the overreaction or reaction of your cells will also have a negative benefit. No, negative that was benefit. So there's something wrong with the words class. Okay, they have a negative effect to the immune to the human host. For example, damage to your host tissue. And also the second mechanism class of your digestion is the formation of your respiratory bursts. I know that the presentation is somewhat intricate and very complicated class, but I wanted to just show the reaction of a respiratory burst. Because the respiratory burst, the main function of this one class is to produce a massive number of your oxygen radicals. Like for example, you have your O2 negative, that your superoxide anions, you have your hydrogen peroxide, and then you have your hydroxyl radicals, and then you have your nitric oxides. Okay, so in immunologic books, if you can observe here, in this illustration is lifted from Abbas. Okay, so you have your here neutrophil. Okay, tapos kakainin niya, diba class? Tapos you have your phagosome. So with your phagosome, aside from the phagosome class, you have your formation of your respiratory burst. This is the preparation here. Pagdating, this is the bacteria, right? Pag nag-fusion na yan sa class, you have your production of your oxygen radicals, right? So you have your hydrogen peroxide, then you have your SOD, and then you have your O2 negative, and then you have your OCl. These are oxygen radicals class that are very toxic to the organism. Okay? And the activation of your enzymatic complex, your NADPH oxidase, then phagosome membrane via to your hexone monophosphate pathway, then we call that one respiratory burst. The oxygen radicals class, example, you have your oxygen that are very highly toxic to your microbes. You have your hydrogen peroxide, an important bacterial agent, okay, but more stable than others. And then you have your HOCl, okay, hypochlorite radicals, a powerful oxidizing agent. Okay, so the main goal of respiratory burst is to produce a large amount of your oxygen radicals that are toxic to your bacteria. Okay, so aside from the granules that we mentioned, we also have this one class, respiratory burst. And after digestion, of course, what's the last part? Dapat, kung may kinain ka, dapat you need to defecate that one. Correct? So, the process is called exocytosis, guys. Okay, residual bodies containing your indigestible materials are excreted as waste. Then, meron tayong mga cleaning cells, like for example, other macrophages. Then they will try to engulf that one kasi ano yan sila, garbage. And then, all are clean with your phagocytosis. So imagine, right? Because before, we just know that, kakainin lang yan ng immune cells. But meron palang mga formation of your phagolysosome to your formation of your phagosome to your lysosome to phagolysosome. Then we also have a respiratory burst that will increase, that will produce a overproduction of your oxygen radicals that are toxic to your bacteria. But take note also, class, that your oxygen radicals is not only toxic to your organism but also toxic to our human body. That's the reason why if you have a a large amount of bacteria, then a large amount of immune cells will also do the phagocytosis, and a large number of your oxygen radicals will also be increased, leading to, like, for example, tissue damage. Then, therefore, we need to limit ourselves for any invasive infection that may activate our respiratory burst. Okay? And that's it, class, for your phagocytosis, kay innate immunity 2. And I hope, and this is the last part of your innate immunity. So I hope that you learned something, because uh, my discussion is on the primary part of our immunity. And then on the next series of lectures, we'll discuss your adaptive immunity. So hopefully that at the end of these video lectures or video lecture series, you're able to get some points that you can understand, or the concept lang man lang, class, na okay, ito pala yung concept ng immunology. Ito pala yung concept ng innate. Ito pala yung phagocytosis. Ito pala yung APRs. Okay? So for now, the main purpose of the first watching this video lecture is to appreciate the beauty of your immunology. And then for the preparation of your quiz, exam, and pre-board exam and board exam, because you have MTAP, then you can, go back again to the video lecture, watch again, and then, understand. Okay? The main primary requirement for immunology to be absorbed, class, is understanding the basic principles. So hopefully that, you get something, and then thank you. And I hope that there are no problems with the understanding the topic and concept. Okay, so thank you, class, for listening with our innate immunity lecture series. Okay, thank you. Bye-bye. Bye, class. Okay, I'll try to, I cannot find my cursor kasi, class. And okay, thank you. Again, so if you have some questions and or clarifications, don't forget to contact your professors. Okay, bye-bye. Bye, class.